Hello everyone, and welcome to another exciting talk in our immuno-oncology webinar series. As you're all aware that PBMC effector cells used for cytotoxicity applications like ADCC have inherent donor variability, thereby reducing their consistency and reproducibility in lot release assays for biologic drugs. Also, ADCC or Antibody-Dependent Cell Cytotoxicity Assays that measure cell death often have low signal-to-background ratios and suffer from this donor variability of the PBMCs. To address the need for robust and reproducible ADCC assays, we at DiscoverX are very excited to introduce the new KILR CD16 Effector Cells that are single donor-derived immune effector cells to measure target cell death in ADCC and bispecific antibody-mediated T-cell redirection applications. These are universal immune effector cells that ensure assay reproducibility for screening, characterization, and QC lot release of antibody drugs. Today's webinar is titled Robust and Reproducible ADCC and T-cell Redirection with CD16 Engineered Effector Cells, which will focus on these new effector cells and is presented by Dr. Jane Lamartine, who is the Director of R&D at DiscoverX. Before we begin our presentation today, I would like to remind you that this webinar is designed to be interactive, and you can submit your questions by typing them in the Q&A box on your screen. All questions will be answered in the Q&A session at the end of the talk. The presentation slides and the recording of today's webinar will be available on DiscoverX's website, and the link will be emailed to you tomorrow. So without further ado, I'd like to hand over to Jane. Jane?
Thank you, Alpina. Good morning, everyone. Thank you for taking the time out of your busy day to join us this morning to hear more about our new effector cells that I'll be describing for you. So I want to begin by putting some context around why we have these cells available as a new product. So bioassays, which is their intended use, are critical components of the development process for biologics, primarily important in lot release and stability testing of drug product, and also in product characterization. Now, from a regulatory perspective, bioassays must serve as a surrogate measure of the biological activity of the therapeutic in vivo. So as such, there's a requirement for the assay to be as reflective of the mechanism of action of the drug as possible. So for class 1 therapeutic antibodies, of which there are many on the market, these antibodies have an Fc domain to the antibody, and that domain can be involved in Fc-mediated effector functions in vivo. And these effector functions result in antibody-dependent cellular cytotoxicity, or ADCC. Therefore, ADCC assays are required during development to quantify and monitor that effector function. So the ideal ADCC assay for lot release with these particular points in mind must be reflective of the drug MOA and therefore measure immune cell-mediated killing, and ideally provide consistent performance from effector cells that eliminate donor variability and minimize lot-to-lot variability. So if we keep those things in mind, that would be the ideal ADCC assay to use in a quality environment. Now the reality, of course, as we know, is quite different. In order to be as close to physiologically relevant as possible, one would imagine using primary cells would be the best opportunity, and many people are using primary cells primarily as characterization assays. But the challenges in working with primary cells are noted here and are very well understood. There is high donor-to-donor variability, not to mention that there can be differences lot-to-lot from different blood draws from the same donor. There are often logistical challenges with obtaining enough PBMCs or NKs during the lifetime of the drug. And then in addition to that, many ADCC assays, especially with PBMCs, often have low signal to background ratios and very shallow hill slopes. So that makes producing reproducible dose response curves for potency very challenging. Now, many groups have gotten around using primary cells by utilizing engineered NK92 cell lines that are overexpressing the CD16 receptor. And this reflects what is normally expressed on NK92s in vivo, but just with a higher expression level and a high affinity variant for the FC portion of the antibody. But the challenges in working with these NK92 cells are somewhat similar to that of primary cells in that they are quite challenging to grow, and varying culture conditions can result in varying results in the effector function of these NK92 cells. But most importantly, what we find is that these cells are not compatible with many target cell types. And indeed, what they do is induce high cell killing, background cell killing in certain cell types, which make them very difficult to use across the board. And then as an additional weight to using these, they are expensive in terms of long-term license fees that are associated with using this particular type of line. So from this perspective, to use a cytotoxicity reflective of the MOA of ADCC in lot release is challenging unless one can identify a reliable source of human effector cells that could address these challenges. So this is why we're very excited about the release of this particular product, which is a single donor-derived engineered effector cells. And we have called these KILR CD16 Effector Cells. These are not an immortalized cell line, but they are primary human cytotoxic T lymphocytes that have been engineered to stably express the V158 variant of CD16. These are manufactured in a GMP facility and using uniform and highly robust SOPs to ensure lot-to-lot consistency over time in producing these particular effectors. So what we're seeing here on the bottom, because these are T cells, are some characterization of the surface markers on these particular populations to demonstrate that these are cytotoxic T lymphocytes. So if you look at the histograms on the bottom, the y-axis on each plot is the surface marker CD8 for cytotoxic T lymphocytes. And on the x-axis, we have different surface markers, either CD4 on the left, CD3 in the middle, or CD16 on the far right. And what you can see is that these cells are 98% CD8 positive and with a very small proportion of CD4 positive cells. If you look at the middle panel, they are 98% CD3 positive. So the CD8 positive cells are predominantly CD3 positive, and 99.7% of the CD8 positive cells are also expressing CD16. So we have strong, stable expression of CD16. So the concept of the utilization of these particular T cells is to use them for ADCC. And the graphic here shows you the concept as we've already described it, in that in vivo, the ADCC is mediated by the NK cells that natively express the Fc gamma R, in this case the CD16, and that engages the Fc portion of the antibody, which recognizes the antigen on the surface of the tumor cell, and this brings the NK cell to the tumor cell and induces lysis. So what we do here with our KILR CD16s is basically mimicking that, but introducing the CD16 into the killer cell, into the T cell. And then it's recognizing the antibody, which also recognizes the antigen on the target cell and induces cell death. So this is functioning essentially as an immunological synapse for killing of the target cell. And this is independent of the MHC1 status of the target cells. So we're really only relying on CD16 here for the specificity of killing. So one question you may ask is why use T cells rather than NK cells, since NK cells do natively express CD16. And I alluded to this earlier, although NK92 CD16 lines are often used as effectors for ADCC and QC labs, there are many disadvantages, and many groups have trouble working with these NK92 lines. They are difficult to grow, and most importantly, they have high non-specific lysis with many cell types. So this is a graphic just showing you an example from two different cell backgrounds. The SCO3 cells are a resistant cell line, so they are not easily killed by NK92, whereas the ARH77 cells, which are on the bottom, are sensitive. So you can see the difference in magnitude if you look at the blue lines, which correspond to killing with the NK92. The SCO3s have very little basal killing, so this is all antibody-independent killing that we're looking at here, whereas the ARH77 have almost 60% basal killing. So this really increases the background in the assay. And the ARH77 data here is reflective of many other cell types that we've looked at, especially suspension cells. Now you'll note that the PBMCs have some background, and that's shown in the gray bars here, but it's nowhere near what you normally see with NK92. But importantly, the KILR CD16 cells, which are shown with the purple bars here, which are very small, have very low background lysis, so even lower than what we see with PBMCs and significantly lower than what we see with NK92. So the specificity of response in an ADCC assay will be driven by your antibody. So how are these cells to be used? So this is meant to complement our existing platform of KILR products, which are cell lines and materials that allow you to make your own cell lines. And the concept, if you're not familiar with the KILR technology, is shown in this cartoon here at the top. So the KILR CD16 effectors are meant to be used as cells that you thaw and rest before you use them in your assay. And you rest them at least 24 hours, but you can allow them to recover for 3 days, 7 days, whatever works best with your workflow. And then you plate your target cells that are, in this case, expressing a reporter protein that's labeled with our beta-gal tag, which is called PL. And you incubate those target cells with your antibody, and then add your KILR effector cells. When the KILR effector cells bind to the antibody and induce killing, that releases our reporter protein that is tagged with our beta-gal fragment EPL. And that's released into the medium. So once it's released into the medium, you can add we add the KILR detection reagents which contain the enzyme acceptor, which is the other fragment of our beta-gal reporter system, and substrate, and then you read that signal on a luminometer. So the nice thing about this assay format is it's all homogeneous. You're releasing protein into the media. There's no need to transfer your supernatant into another plate. There's no spinning or any of those types of steps that might be involved in other types of ADCC readouts. It's all done in a single plate. Now, the KILR effector cells that we're speaking about today are compatible with our KILR assays, of course, so that you can use target cells developed from our menu of over 30 different cell models. You can generate your own target cells using our KILR reporter, using our retroparticles, or you can use your own in-house target cells and cell lines that you've developed for your own ADCC assay readouts. So they're compatible with basically any type of ADCC assay as long as it is an immune effector-mediated readout. So from that perspective, assays that are compatible with these effectors would include the KILR ADCC chromium release, europium, calcium release, even cytox glow, those types of readouts. So some highlights, as I just mentioned, the KILR effectors are compatible with many different cytotoxicity assays, such as the chromium release, calpain release, etc. They are not compatible with binding assays. So it's anything that uses an effector-mediated killing mechanism. The important thing, and we'll show you some examples here, is that these particular cells eliminate donor-to-donor variability because they are derived from a single donor. They are easy to implement in any lab because they are supplied as a frozen, ready-to-use cell, although one needs to allow them to recover. Just from a physiological perspective, these are T cells that need some recovery time before you can use them in your assay. But they don't require complicated culturing. They are fit for long-term QC testing in that we have observed high lot-to-lot reproducibility of the effectors, and we'll show you some data for that. And finally, they're relevant measures of ADCC, and we've also shown that they can be used for T-cell redirection, and we'll show you that data as well. So one of the benefits of using these particular effectors is that they actually produce a larger assay window than what we typically see with PBMC. So the data that I'm showing you here, there are two different models that we've tested in this slide. The data on the left is our CD20 model for rituximab, and the one on the right is our trastuzumab model. So if we focus on the data on the left at first, what we've compared here are the KILR effector cells, the CD16 effector cells, in pink versus PBMCs. These data were generated in the same experiment. And what you can see is that although what we typically use are PBMCs at a ratio of 25:1, we've already reduced the KILR effectors to a ratio of 10:1, and yet we still see over a five-fold difference, or roughly a five-fold difference in signal to background, and a two-fold increase in the max cytotoxicity relative to PBMCs that are, in this particular case, also from a VV donor. And yet the change in EC50 is still quite small. What you can see on the right is a more difficult to kill cell model, which is our SCO3 cell model. Here what we've observed is that with PBMCs at a ratio of 25:1, we get a very shallow hill slope, and with PBMCs, but we see a much better response in killing with the KILR CD16 cells using a 12.5:1 ratio. And again, the EC50s are within twofold, but the signal to background and max cytotoxicity are significantly better when you use the KILR CD16 effector cells. So the important point here is you already have an advantage in using a smaller target effector to target ratio, and you can adjust that ratio even further as you optimize your assay with these new effectors. So as we mentioned, these cells are can be used in a thaw and use environment. So what we tested was the ability of the killing capacity. How does that perform after you thaw the cells? So what we have here are data from thawing and letting them recover for 1 day, 3 days, 7 days, or 10 days. And what you can see from the graph and also from the data on the right is that you get very highly reproducible cell killing in terms of the data and the recovery even after 10 days. So there's little difference in the killing capacity, whether you use them 1 day after thawing or whether you use them 10 days after thawing. But you do need to allow them to recover at least 24 hours in order to use them. And what you can see here is from the EC50 change is less than 5% relative standard deviation in EC50. So if we look at inter-assay variability with these effectors versus PBMCs from a VV donor or versus NK92 CD16 effectors in our SCO3 assay, what you can see is that although the EC50s are slightly different between using the different effectors, the actual variability in the assay is much lower for the KILR CD16 cells, which are shown here in the pink, where we see a 9.6% RSD for 3 different experiments. If we look at the PBMCs, as you would expect, although the EC50s are lower, the actual range in EC50s is 42% RSD, whereas the NK92 is somewhere in between at 19.5%. So again, we see much tighter data with the KILR CD16 effectors. Perhaps most important is that we observe excellent interlot reproducibility. And these are data generated from 4 different lots that were manufactured over the course of 2 years. And you can see that the performance in a rituximab model is quite similar. What we're observing is lot-to-lot differences of less than 35% RSD, which is lower than what you would typically see when you look at intradonor variability for PBMC. And unfortunately, this particular cell background is not compatible with NK92, so we don't have data directly comparing that. But nevertheless, what we see is quite good, a low lot-to-lot variability. So I wanted to switch gears a little bit and talk about other potential applications of the KILR CD16 effector cells. And so one of the questions that we have received from clients is, could it be used for T-cell redirection? So T-cell redirection can be done with a number of different types of antibodies. The one that we chose to evaluate is a BiTE-style molecule, blinatumomab, that has 2 different targets. The first is the CD3 antibody end that binds to and activates a T-cell. And the other end of the bispecific recognizes CD19 on the surface of B cells. And so we use this particular antibody and the KILR CD16 cells in our KILR model. What we've shown here is that we have pre-incubated the CD19 expressing cells with the antibody, and then introduced the KILR CD16 cells, and we're measuring release of our KILR protein into the media. So we evaluated this particular molecule initially in pan T cells. So these are T cells that have been isolated by negative selection and evaluated with our KILR Raji cell model. And what you can see here is, in the absence of any effectors, you have very low cytotoxicity in this model, as shown with the teal dots. But a 24-hour incubation with blinatumomab in the Raji model produces a nice dose-dependent response with the pan T cells. So if we compare that to the KILR CD16 cells, what you can see from a time course here is that the kinetics are much faster than what we typically see with pan T cells. And the optimal response that is comparable to what we saw with the 24-hour incubation with pan T cells is observed in 6 hours with the KILR CD16 cells. Importantly, the EC50 that is reported for blinatumomab in the BLA is between 10 and 100 picograms per mil, and both of these assay formats produce an EC50 in that range. It's slightly more potent with the KILR CD16 effectors, but nevertheless within the range that's expected and is physiologically relevant. So to conclude, our KILR CD16 effectors are capable of driving robust and reproducible effector cell performance for cytotoxicity assays, not only ADCC, but also T-cell redirection. These are single donor-derived primary human effector cells, and the benefit of using these, obviously, is to eliminate donor variability from your ADCC assays, to produce results with high reproducibility. These are effector cells that are primary cells, but they are engineered to stably express CD16, as I showed you, and that CD16 is a requirement for mediating ADCC. We have observed high lot-to-lot reproducibility that enables long-term support of QC lot release bioassays, and it drives and measures cell death of target cells, which is the reflective of the true mechanism of action of ADCC, which would be ideal for an MOA-based assay. They are cryopreserved, ready-to-use cells, but they do require at least a 24-hour post-thaw recovery period before using them in your assay. But with that, there is no complicated requirement for propagating them. You just add them to the media and let them recover until you're ready to use them. So that's quite QC-friendly in terms of adoption in any workflow. And finally, we observe higher signal-to-background ratios with these effectors that should improve your performance relative to PBMCs if you are using them either for lot release or characterization. So the KILR CD16 effectors are sold as either a single vial or a 5-vial pack, and there is accompanying cell culture media and IL-2 required to propagate them. And so those are all provided on our website as supporting products. From the perspective of what you receive when you purchase the cells, they're typically shipped as 20 million cells per vial, plus or minus 15%. Each of these vials will come with a certificate of analysis and a user manual. Finally, the cells are shipped on dry ice, obviously. These are primary cells that are cryopreserved. It's very important that they be transferred to the vapor phase of liquid nitrogen when you do receive them because they are sensitive. But beyond that, they are quite straightforward to work with. So with that, I'll close and ask if there are any questions that you have, and thank you for your attention.
Thank you, Jane, for the presentation. Before we begin our Q&A session, I would like to remind our attendees that if you have any questions for our speaker, please submit them in the Q&A box on your screen. So let's look at the questions that we have. Why are these effector cells more efficient in T-cell redirection assay? Jane?
There could be a number of reasons why. One possibility is that these are 98% cytotoxic T lymphocytes, as opposed to pan-T cells, which are a population of CD4 and CD8 positive cells. And the proportion is quite different. So it's only typically about a third of the cells in the pan-T cells would be CD8 positive. So it could just be a potency-related issue in terms of the proportion of CD8 positive cells.
Thanks, Jane. The next question is, how does the killing profile of the CD16 effector cells compare to those of the NK cells for ADCC?
Well, we haven't compared them one-to-one, but our experience with NKs relative to PBMCs is they should work just fine. And the ratio of, if we compare to NK92, the ratio of NKs would be very similar to what you would use for the CD16 effectors, and you might be able to decrease that even further, but that's probably what I would start with, is a comparable ratio to what you use for NKs.
Thank you, Jane. The next question we have here is, are these effector cells single use only, or can these be propagated for a longer time?
So The cells themselves do not, they don't have a lot of proliferative capacity, but they can be maintained in culture for up to 2 weeks. So, what we recommend is you can certainly use them right away in that after 24 hours. You can use them on day 1, or you can, thaw them and use them after a weekend rest or after 7 days. It's really reflective of your particular workflow, what works best for you. We internally have used them within 24 hours or let them rest over the weekend and then use them.
So in the next question that I see here is, in your last but one slide where you're listing the products, you did not list any antibiotics. Do you not need any antibiotics to grow these cells or maintain these cells if you're keeping them for such a long time?
No, we do not need antibiotics. And it's not intent, you cannot propagate them for weeks on end. They are T cells, they're only going to survive for a small amount of time. And we do maintain them in the presence of IO2.
Okay, thanks, Jane. For the NK cells, you mentioned the ET ratio. So for PBMCs, NK92, what are your recommendations for different cell models?
Well, it's going to be very dependent on whatever your assay is. So I think what you would do, our experience is with PBMCs that we would typically reduce the ratio to, let's say, for example, we're using a 25-to-1 ratio for PBMCs. We can use anywhere from a 5-to-1 to a 10-to-1 ratio. So you just would optimize within that range and find what works best for your particular cell model. If you have a difficult-to-kill cell model, then you might need to use a slightly higher E to T ratio than you would for something like the Rajes.
Thank you, Jane. We have time for one last question. Are there any types of cells that will not work with these effector cells?
Meaning target cells?
I think it would be target cells.
Yeah. I'm not aware of any. Obviously, we haven't tested every possible target cell, but of the ones that we've tested, we haven't seen anything that couldn't be killed by ADCC if you have the antibody recognizing the antigen on your target cell.
Okay, great. Thank you, Jane, very much for the great presentation and your feedback on different questions. So to our attendees, I'd just like to remind you, the recording of today's webinar and the slides will be made available on the website shortly, and we will be sending you the link to the download. So I'd like to thank you everyone for joining today for the webinar, and if you should have any questions, please feel free to email your relative technical support teams in US or North America or in Europe. We'll be very happy to respond to your responses if you have any questions. So thanks everyone, and have a great rest of the day. Bye-bye.